Hello guys, welcome back to my YouTube channel. This is a Life Sciences Grade 12 lesson. We are continuing from the previous lesson, which was on the structure of the cell, revision structure of the cell. So these lessons are a build-up. So if you did not check out that video, please pause this video, go to that video, do me a favor, like and subscribe. Feel free to comment, feel free to share the link to this video because it would mean that more people will be able to gain access and ensure their academic excellence in Life Sciences Grade 12. My name is Tumi Selani. I am a teacher at Milner High School here in the Northwest Province. This is lesson number 2. It is titled The DNA Structure. Okay, the lesson breakdown. Okay guys, now here we are looking at the lesson series breakdown. I've broken down the lesson series into 7 lessons. Each lesson will be on a separate video. So, on lesson 1, we'll dealt with the revision of the structure of the cell. So be sure to check that video out. In this lesson, we looked at the different structures and the different functions and the different organelles that we find in the cell. Okay, this is important for you to understand because as we will be doing with the different processes in DNA Code of Life, it is important for you to understand the locations within the cell in which each process takes place. On this lesson, which is lesson number 2, we will be dealing with DNA structure, understanding what DNA looks like, the origin of DNA, and how DNA functions. On lesson 3, which will be on a separate video, we will be looking at DNA replication. On lesson 4, we will be looking at the history of the discovery of DNA. So it is quite interesting but very controversial. On lesson 5, we will be looking at a different nucleic acid, which is RNA, its structure, how it functions, and the different types of RNA. On lesson 6, it is going to be a 2-part lesson, which is protein synthesis. On the first part, we will be looking at the process of protein synthesis, which is divided into transcription. I am very sorry for that. There is supposed to be transcription, movement of mRNA, as well as translation. The second part of lesson 6 will be on amino acid coding. This topic is a very problematic one because every single year at the Marking Center, they compile what we call a diagnostic report. So on the diagnostic report, amino acid coding seems to be a recurring problem every single year. But don't worry, I'm going to make it very easy for you in order to ace this. Then on the last video, which is going to be lesson 7, we are going to be looking at DNA profiling. Okay? Now, we will be looking at the definition and uses of DNA profiling. We will be looking at factors that affect the reliability of DNA profiling. And then I'm going to give you skills in order to interpret different DNA profiles in order to determine paternity, determine who a biological father of a said child is. And then lastly, we will be doing a little bit of investigations into forensics in looking at different DNA profiles, to determine a possible criminal in a criminal case. Okay, so that is the lesson background. Let us get right into the lesson itself. Okay, now DNA is an abbreviation. Full name is deoxyribonucleic acid. I know it's quite a long term, but let's just break it down into syllables. Okay, deoxy, that's the D, ribonucleic, N acid. So deoxyribonucleic acid. The more you say it, the more you'll be able to pronounce it, okay? So say it more and more and more and more, and you'll build a memory bank in your tongue. It will become easier. It will just slide off of your tongue. So the first slide, or the first couple of slides, we will be looking at the introduction of this deoxyribonucleic acid. So, DNA, introduction. What is DNA? I'm sure DNA is a term that you hear often about, okay? But a lot of people don't actually know what DNA is or what it does. So, you as a grade 12 life sciences learner, you are required to know what DNA is and how it functions. That's the subject of today's lesson. So what is DNA for 2 marks? So this definition that I'm going to give you acts as a definition and as a function as well. Okay? So the answers to this question is DNA is a nucleic acid molecule. It's a nucleic acid molecule. So it is a molecule that is found in the nucleus, nucleic acid. Hence it is deoxyribonucleic acid. All right? So DNA is a nucleic acid molecule that is mainly responsible for. Let me just erase there. There we go. It is mainly responsible for carrying the genetic code for protein synthesis. So DNA has a specific code, okay? A genetic code. It has a code for protein synthesis. Okay, now, what is protein synthesis? Cells have the ability to make their own proteins. So, synthesis or to synthesize simply means to make, to manufacture. Okay, so when you are making something, we say that you are synthesizing that particular thing. Now, what is it that we are synthesizing in this case? We are synthesizing, or we are making proteins. Okay, so cells have the ability to make their own proteins. But at any given time, cells would actually make a specific protein. They are required to make a specific protein. Now, the instructor to tell the cell which protein is to be made is DNA. So DNA instructs the cell to make which proteins at any given time, and that instruction comes from the code that is in DNA. So DNA has a specific code that would tell the cell which protein to be made. So the first function of DNA is that it carries the genetic code for the production of proteins. I hope that is clear enough. And then, second main function of DNA is that it carries hereditary information from parent to offspring. From the previous lesson, we have explained what hereditary information is. Now, hereditary information, okay, hereditary is derived from the word, what is this word? Inheritance, yes. Inheritance, okay? Now, all our characteristics, whether you have blue eyes, brown eyes, the shape of your eyes, the size of your feet, your skin tone, your blood type, the shape of your nose, the length of your nose, the texture of your hair, etc., etc. All these characteristics, all of them are coded in DNA. So everybody's DNA has a specific code that codes for these characteristics, okay? We call those genes. Now, where are they stored? The code for these characteristics are stored in a person's DNA. Now, who we are is a result of, or our characteristics, we have inherited them 50% from our mothers and 50% of our fathers. Okay? So these codes for our characteristics are stored in DNA. Now, the DNA of your father's sperm contains his genetic information, this hereditary information of his height, his eye color, shape of the nose, whatever. So all his characteristics are stored in his DNA. The DNA that is found in the sperm, okay? And the DNA of your mother contains your mother's characteristics in her DNA, the DNA that is found in her egg cell. We know that during fertilization, the sperm of the mother, the sperm of the father and the egg cell of the mother come together. They fuse. So essentially, we are fusing the DNA with the characteristics of the father and the DNA of the characteristics of the mother to make us. Okay? So the DNA contains hereditary information of the father in its sperm cells. And then the DNA of the mother carries her hereditary information in her egg cell. So the hereditary information of our parents, are passed from them in their DNA contained in the sperm and the egg cell to us, the offspring. I hope this makes sense. All right? Now, let me just erase this. So to go back, DNA is a nucleic acid molecule that is mainly responsible for, it carries the genetic code, so the information for which protein is going to be made. So it is carrying the genetic code for protein synthesis. Secondly, it carries hereditary information from parents to offspring. So it takes the hereditary information from mom and dad and passes it on to us, the offspring. So essentially, this is how DNA looks like. Like we said, this acts as both a definition for DNA, so it describes what DNA is, and it also describes what DNA does. So whenever you're asked for the significance, function, or importance of DNA, this is what you ought to provide. If you are asked for the definition of DNA, this is what you may provide as well. So it's a 2-in-1 special. The following question: where in the cell is DNA found? So this question is asking for the location or the site of DNA. All right? Now, in the animal cell and in the plant cell. In the animal cell, there are 2 locations where DNA is found, and in the plant cell, there are 3 locations where DNA is found. Remember, from the previous slide, from the previous lesson, we have studied briefly the different structures of the cell that are relevant for the grade 12 curriculum. So if by any chance you have no idea what we are talking about by the different organelles from where we find DNA, please go back to the previous lesson and revise the structure of the cell. It's very important for your grade 12 journey. Right. Now, most of the cell's DNA is found in the nucleus. So it is both the animal cell and the plant cell have nuclei. Remember, nuclei plural and nucleus singular. So we have one nucleus, many nuclei. So most of the cell's DNA is found in the nucleus. And this DNA we refer to as nuclear DNA. Now, DNA that is found in the nucleus is referred to as nuclear DNA. So we find it there in the plant cell and there in the animal cell. Okay. The second location, some is found in the mitochondria. Okay. Where is the mitochondria? There is the mitochondria. There is a mitochondrion 1 in the plant cell, and there's another mitochondrion 1 in the animal cell. Remember, one mitochondrion, many mitochondria. Now, the DNA that is found in the mitochondria is called mitochondrial DNA, or it is abbreviated small m, small t, capital DNA. All right. The last place where we find DNA. Plant cells also contain DNA in their chloroplasts. This is known as chloroplast DNA. So remember, animal cells do not have chloroplasts. Now, only plant cells will contain chloroplast DNA. Why? Because they have chloroplast. So there is that note. Animal cells do not have chloroplasts. Therefore, DNA is found in 2 organelles of an animal cell, but in 3 organelles of a plant cell. So in plant cells, we find DNA in the mitochondria, nucleus, and the chloroplast. But in animal cells, DNA is only contained in the nucleus and the mitochondria. Okay. Continuing with DNA. The next question is describe and explain. Describe and explain the shape of DNA. Now, the word describe, guys, the word describe. When you are describing, you are giving the features of something. So, if something is tall, dark, and handsome, why am I using that example? Okay. If they say, give the description of Thabo, you are saying Thabo is tall, dark, and is handsome. You are telling us the features of Thabo. So that is what the word describe means. Just tell us about the features, nothing else. And an explanation would be, why is Thabo tall, dark, and handsome? So explanation, you give details to the structure. Okay, so please mind these, what do they call them? These descriptive terms in the questions. When they say describe, you just provide the features of that particular structure or that particular process or whatever they'll be asking about. If they say explain, they mean give detail as to why. So we need further details. So this question is asking us to describe and explain the shape of DNA. So the description, let's start with the description. So the description is that DNA is a double helix. That is the description for 2 marks. It is a double helix. Remember, describe means tell us the features of it. Now, to explain what double helix means. Double means, the double part means that it has 2 strands. Okay? Double means 2 what? 2 strands. So DNA has 2 strands. What does the word helix mean? Helix means it is twisted. So double helix is a scientific term used to describe the shape of DNA. The double part means that it has 2 strands, and then the helix part means that these two strands of DNA are twisted. All right, so there is where we get our 4 marks. Okay, let me just erase and let us visually see what we are talking about. So, description. The double part, remember, means that DNA has two strands. Here is, so this is one strand of DNA, and then there is the other strand of DNA. So DNA has two strands, okay? Strand 1 and strand 2. But we also need to accommodate the helical part. So something that has a helical shape means that that shape is twisted. So this DNA does not only have two strands, but these two strands are twisted. So this is what we call a double helix. So there we go. There is our DNA structure. It has two strands, and those two strands are twisted. Hence, double helix. Okay, now that was the introduction of DNA. We are going to look now into its structure. How is DNA built? All right, let's get right into it. Okay, guys, now in this slide, we will be describing the structure of DNA. How is it built? Okay. DNA is a relatively long molecule. When you uncoil DNA from a chromosome, it actually is around, some sources say, around 4.9 meters, 5 meters, some sources say 7 meters long. So it's quite a long molecule, relatively large. 7 meters is quite long. Okay. Now, any molecule that is that long has to be made up of smaller subunits. It has to have a building block. Imagine a house. No? Now, a house doesn't come built and it's just implanted. It's something very large. So you have to build a house. Okay? So how do we build a house? There are smaller subunits that we call bricks that are packed on top of one another and next to each other to eventually build that large house. So DNA being a long and large molecule, it needs to have its own form of big bricks, its own form of building blocks. Okay, so DNA does not come as a large molecule. It is built to become as large as it is, just like in the case of a house. So, the building blocks of DNA. So, the word monomer means a building block. Okay, just like a monomer of a house, if I'm to use that analogy, the building block of a house is a brick. The monomers, the single units, the building block of a DNA molecule is a nucleotide. Okay, so DNA is made up of monomers or building blocks called nucleotides. So the word the word monomer means a building block. So a monomer is a single unit of a large molecule or a compound. All right? So it is a single unit of a large molecule or a single unit of a compound. A monomer is also often referred to as a building block. So the analogy, just like a brick building block is stacked onto another brick to build walls that would eventually form a house, Nucleotides are also stacked together to build two strands of DNA. Like I said, DNA being a large molecule, it cannot just exist as being such a large molecule. Oh my God, my dogs are making so much noise. Excuse the noise of the dogs, guys. Okay, they have calmed down, hopefully. All right, so like I was saying, DNA being a large molecule, it cannot just exist as being large. It needs to be built to be large, okay? So it is built from building blocks that are called nucleotides. So the term monomer means a single unit, a building block. So those monomers are called nucleotides. All right? So the analogy of building a house, brick, 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 eventually this will form a house. So in the case of DNA, we would have a monomer stacked on top of a monomer, on top of a monomer, on top of a monomer, a nucleotide stacked on top of a nucleotide, stacked on top of a nucleotide to make the large molecule that is DNA. So one nucleotide, so one brick or building block has three components. It has three parts. Okay, follow me here. So DNA is made up of building blocks that are called nucleotides. They are stacked and stacked and stacked to make the large molecule that is DNA. So that one building block, that one nucleotide has three components or three parts. Firstly, it is made up of a phosphate group. Okay, so the first component or the first part of a nucleotide is a phosphate group. Please do not just say phosphate, guys. Do not just say phosphate. We are not referring to a single element, phosphate. It's a group of phosphates. Okay, so phosphate group. I know that some publications or some learning resources say phosphate. Please, phosphate group. Okay, and it has a sugar portion or a part that is made up of sugar. There we go. There is our sugar portion. So it is the sugar portion and the phosphate group. And then the last component is what we call a nitrogenous base. Very careful here again. Not nitrogen base, not the element nitrogen. The base contains nitrogen. So please, let's use nitrogenous adjective, nitrogenous base, not nitrogen base, not the noun nitrogen base. The adjective nitrogenous base, a base that is nitrogenous. Very careful. Phosphate group, or you can say phosphate portion. A sugar portion and a nitrogenous group. Not phosphate, not nitrogen base. Please be very careful there. Okay, so the first component is a phosphate group. Then we have a sugar part or a sugar portion. And lastly, we have a nitrogenous base. I'm going back. DNA, being a large molecule, is made up of small building blocks. Those building blocks are called nucleotides. So this is one nucleotide. This whole thing is one nucleotide. But this one nucleotide is made up of three components or three parts. We have a phosphate group being one of them. We have a sugar portion. And lastly, we have a nitrogenous base. All right? I hope you follow. Okay. It gets a little bit more complex. In the nucleoplasm, we have nucleotides that contain, we have four different types of nitrogenous bases. Okay? I repeat, I'm going back again. I'm sorry, but I need to go back again. Remember, we are describing the structure of DNA or how DNA is built. All right? Now, DNA, being a large molecule, is made up of smaller units or monomers that are called nucleotides. One nucleotide, this whole thing is one nucleotide, is made up of 3 parts. The first one is a phosphate group over there, and then a sugar portion over there, and it may contain one nitrogenous base over there. It must contain one nitrogenous base. Now, the nitrogenous bases, there are 4 kinds of nitrogenous bases, but one nucleotide carries one nitrogenous base. But this nitrogenous base can either be adenine, abbreviated A. It can be thiamine, abbreviated T. It can either be cytosine, abbreviated C, or guanine, abbreviated G. Okay? Now, phosphate group, sugar portion, nitrogenous base. So the nitrogenous base can either be, remember, one nucleotide carries only one nitrogenous base. So that nitrogenous base can either be adenine, thiamine, cytosine, or guanine. All right? So this nucleotide, there we go, would have a phosphate group, a sugar portion, and adenine as a nitrogenous base. Or it can have a phosphate group, a sugar portion, and thymine as a nitrogenous base. Or it can have a phosphate group, a sugar portion, and cytosine as a nitrogenous base. Or it can have phosphate group, a sugar portion, and guanine as a nitrogenous base. All these 4 things are nitrogenous bases. So one nucleotide can only contain one nitrogenous base. Can either be adenine, thymine, cytosine, or guanine. Those are the 4 nitrogenous bases that we get for DNA. So this is a diagram showing the general structure of a nucleotide. General structure: phosphate group, sugar portion, nitrogenous base. And the nitrogenous base can either be one of these 4. All right. So there we go. Each nucleotide comprises of either one of the listed bases. So it can be either one of these. Not all of them, not 2 of them, not 3 of them, just one. Now, DNA is not the only nucleic acid in the cell. We have another nucleic acid, which is RNA. So RNA, which we will later learn about in later lessons, is also a nucleic acid. Therefore, it means that there is a specific nucleotide that makes RNA, and there's a specific type of nucleotide that makes DNA. Now, when you're asked to describe or to draw a DNA nucleotide, specifically, what must it contain? DNA nucleotides contain, remember, it has a phosphate group and it has a sugar portion. The specific type of sugar that makes up DNA is a ribose. I mean a deoxyribose sugar. The reason why DNA is called deoxyribonucleic acid is because, one of the reasons is because its sugar is deoxyribose sugar. Okay? The molecule is called deoxyribonucleic acid. Deoxyribonucleic acid. The sugar portion contains the specific sugar that is called deoxyribose. We stop there. Sugar. All right? And there is a nitrogenous base that is specific to DNA. RNA contains cytosine. It contains guanine as well. It contains adenine as well. However, RNA does not contain the nitrogenous base thymine. So thymine is only found in DNA. Remember, we have 4 nitrogenous bases. We have cytosine, which we have both on DNA and RNA. Guanine, that we both have on DNA and RNA. Adenine, both found in DNA and RNA. But thymine is exclusively found in DNA and DNA alone. All right? So a DNA nucleotide would contain a phosphate group, okay, as one of its components. And on its sugar portion, you must specifically say deoxyribose sugar when you are describing a DNA nucleotide. Deoxyribose sugar. And remember, in terms of the nitrogenous bases, a DNA nucleotide would only contain 1 nitrogenous base. So the nitrogenous base that we exclusively find in a DNA nucleotide, not in RNA, is thymine. Okay, so look at the title or the heading of this nucleotide. A diagram showing the structure of a DNA nucleotide. So a DNA nucleotide would consist of a deoxyribose sugar, specifically deoxyribose, and it may contain thymine as a nitrogenous base. Okay, so it may be adenine, it may be guanine, it may be cytosine, but thymine is only found in DNA nucleotides, not in RNA nucleotides. All right. Okay, guys, continuing with DNA structure, what you need to note is that the number of adenine bases equals approximately the number of thymine bases. Okay, so if there's, for instance, 1,000 adenines in the nucleoplasm, there will roughly be 1,000 thymine bases as well, and the number of cytosine bases equals the number of guanine bases approximately. So if there is around 2,500 cytosines, there will be around 2,500 guanines as well. This is important to note. Why? We therefore say that adenine is complementary to thymine and cytosine is complementary to guanine. So remember that we said DNA has 2 strands. We said that DNA has 2 strands. It would be strand 1 and strand 2, and then in the middle there we would have our nitrogenous bases, and this side also we would have complementary nitrogenous bases. Okay, so This is what we mean by complementary. If on one strand you have an adenine nitrogenous base, and on the other side you would have a thymine nitrogenous base. Why? Because like we said, where is it? Like we said, adenine is complementary to thymine, okay? And if you have a cytosine nitrogenous base on one strand, the opposite strand or the complementary strand would have a guanine. Why? Because we said cytosine is always complementary to guanine. All right? Okay. So, whenever you see adenine on one strand, on the opposite strand, it must be complemented by thymine. And if you see cytosine on one strand, the complementary nitrogenous base would be guanine. All right? Okay. Now, the two strands of DNA are joined together by weak hydrogen bonds. These bonds join the complementary bases of the opposite strands together in a specific way which forms a code. All right. So, let's check. Now, the weak hydrogen bonds. Remember, this is, excuse me for a moment. This is the first strand of DNA, the left strand of DNA, and this is the second strand of DNA. All right? The statement says that the two strands of DNA are joined together by weak hydrogen bonds. So the weak hydrogen bonds you would find there in between the nitrogenous bases of opposite strands of DNA. All right? The statement continues to say, these bonds join the complementary nitrogenous bases, as you can see over there, of the opposite strands together in a specific way that forms a code. So a code, for example, ATCG and ATCG. This is a code. This is a code for this portion or this piece of DNA. Remember, DNA is quite a long molecule. Okay, so what we see over there, the sequence of nitrogenous bases, ATCG, ATCG. This is a specific code that we find in this DNA. Excuse my handwriting. I am not using a pen, but I'm using a mouse to write, so it's a bit inconvenient. Right, so this is a code of this piece of DNA. Okay. Continuing. See an illustration of complementary base pairing in DNA below. So like we said, adenine is always complementary to thymine, and cytosine is always complementary to guanine. All right? Therefore, whenever you see a T on one strand, the complementary strand, you will find an A on the opposite side. Whenever you see a C, such as there, on the complementary strand, you will see a G. And then the two strands are joined together by the weak hydrogen bonds that connect the complementary base pairs. Okay. Now, how do we make this easy for you to remember complementary base pairing? How do we make it easy for you to remember? So let's just have a quick analogy. Let's use this analogy to make you remember this easier. The apple A, which stands for adenine, always falls from the tree, T. Apple A always falls from the tree, T. Therefore, A, adenine, is always complementary to T, thymine. All right? And the car, C, for cytosine, always parks in the garage, G, for guanine. The car, C, always parks in the garage, G. So this means C is always complementary to what? It's always complementary to G. I hope this makes it easier for you to remember. Okay, we are not yet done with the structure of the cell. The structure of DNA. Let's look at a visual of these things that we have been talking about. Okay, there we have a DNA molecule, strand 1 and strand 2 of the DNA molecule. So you see all these different things over here. Let's just go back and remind ourselves what these things are. Okay, so this is one strand of DNA, and then that is another strand of DNA. Remember, we had said that DNA is a double helix, therefore it has two strands. So in this instance, it is not twisted for the convenience of teaching. It is straight, but remember, DNA has two strands. Okay, now let's check the different components of DNA. So there in the middle, what is that? That is joining one strand of DNA to the other strand of DNA. What did we say that was? We said those are weak hydrogen bonds. Okay. In the older syllabus, you needed to know how many bonds, weak hydrogen bonds, bond adenine and thymine, and how many bond guanine and cytosine, but that is not in your syllabus, that's not in your curriculum now, so you don't need to know how many weak hydrogen bonds, how many bonds of weak hydrogens are required to bond which complementary pairs. So all you need to know is that the two strands are joined by weak hydrogen bonds. The number, you don't need to know that, even if it appears on some publications, but for examination purposes, it is sufficient to know that the two strands are joined by weak hydrogen bonds. Okay, now, what is that? What is that we see over there? So here is adenine. And remember, what is adenine? Adenine is a nitrogenous base, okay? Because it is labeled as A, you need to specify that it is adenine. All right? And then there, what do we have there? Remember, in a nucleotide, we have three components: a phosphate group, a sugar portion, and we also have a nitrogenous base. So we have specified that this nitrogenous base is adenine, and nitrogenous bases are always attached to the sugar portion. Okay, now because this is a DNA molecule, you need to be specific to say this is deoxyribose sugar. All right, if it was an RNA molecule, we would specify the type of sugar that is found on its sugar portion. But with RNA, we'll look at the structure of RNA at a later stage. Okay, now lastly, on our nucleotide, we have that part that is not attached to the nitrogenous base, and that component of the nucleotide is a phosphate group. Just to reiterate, please guys, and a nucleotide contains three components: a phosphate group, a sugar portion, and a nitrogenous base. All right? Now, the nitrogenous base is always attached to the sugar portion, never to the phosphate group. Okay? So, because this nitrogenous base is labeled as A, if they say label this, you need to specify to say it is adenine. And because this is a DNA molecule, we are studying the DNA molecule, you need to specify the type of sugar that's on the sugar portion to say that it is deoxyribose. And the phosphate group would be the remaining one. All right. Now, there we have a structure that we need to label as well. It has 1, 2, 3 components. So there is our nitrogenous base, which is thymine. There would be our deoxyribose sugar, and there would be our phosphate. So this whole structure, what is it called? It is called a nucleotide, specifically a DNA nucleotide. How do we know that it is a DNA nucleotide? Because of this thymine. Remember, thymine is found exclusively on DNA nucleotides and not on RNA nucleotides. All right. Notice how the sugar portions and phosphate groups form the backbone of the strands. Okay, let's check. Let's use the left strand as an example. The nitrogenous bases are on the inside of the strand. So the nitrogenous bases there, thymine, adenine, guanine, thymine, guanine, are on the inside. But the backbone check, the backbone there. Let's check what we have. This is a phosphate group, sugar portion. 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 Therefore, we say that the backbone of each strand forms a sugar phosphate backbone. Remember, a nitrogenous a nucleotide contains 3 components: a phosphate group, sugar portion, and a nitrogenous base. But because the phosphate group and the sugar portion are on the backbone of the strand has a sugar phosphate pattern. Therefore, we say that it is a sugar phosphate backbone. All right? The second point that we need to note, the bases form the inner stairs of the DNA molecule. Okay? So on the inside, we have the nitrogenous bases, and then at the backbone of the strands, we are going to have that sugar phosphate backbone. Another point that we need to note, the nitrogenous bases always attach to a sugar portion, never a phosphate group. I have mentioned this. So always the nitrogenous bases, if you can check, nitrogenous base is attached to a sugar portion. There, nitrogenous base, sugar portion, nitrogenous base, sugar portion. The nitrogenous base never bonds with the phosphate group. And then weak hydrogen bonds join the 2 strands of DNA by attaching the complementary bases to opposite strands together. So the weak hydrogen bonds, as you can see over there, so there is a weak hydrogen bond there. So the weak hydrogen bonds join the 2 strands of DNA together by joining the nitrogenous base, which are on the inside of the strands, as opposed to the phosphate group and the sugar portion, which are on the background. Okay, all right. Now, So on the slide, we are going to look at a simulation again to check how DNA is structured and how DNA actually functions. All right. That DNA is a long molecule, relatively long molecule, which has 2 strands, and then each strand is made up of nucleotides, and the nucleotides contain a code in the form of a sequence of nitrogenous bases, as we can see over there. And because DNA has 2 strands, the sequence of nitrogenous bases are complementary. We said adenine is always complementary to guanine. Adenine is always complementary to thymine, and guanine is always complementary to cytosine. Remember, let me just fast forward for a moment to the part that I need. There we go. Now, remember our analogy. The car always parks in the garage. Therefore, C is always complementary to G. And the apple always falls from the tree. Therefore, A is always complementary to T. So if in one strand there's a G, on the complementary strand, there will be a C. If on one strand you see a T, on the complementary strand you will see an A. All right? So that's how complementary base pairing works on DNA. All right, guys? And something else to note, that the 2 strands of DNA are joined together by weak hydrogen bonds. So in between the complementary nitrogenous bases, we are going to find weak hydrogen bonds. We will explain when we do DNA replication and transcription why these need to be weak. All right? And another thing that we must note, that we've already talked about, that is of particular note, is that the reason why C and G are complementary and A and T are complementary is because, like we said in the previous slides, there is approximately the same number of C's to G's, and there is approximately the same number of T's to A's in the nucleoplasm. All right, let's move on.
And a little bit about how DNA works.
Okay, we have reached the end of our lesson 2, which was based on the DNA structure. I hope that it made sense for you. If there are certain concepts that you do not understand, please go back and rewatch the video or the segment of the video that you do not understand. I am going to read all your comments. So if there is any clarification that you need, please post it on the comments and I will gladly reply to your comments. All right, guys? But as per usual, I am not just going to leave you. We are going to do a few assessment questions to be able to know for sure that you understood the content in this episode. All right. Now, remember, I'm going to be going through question by question. So Pause the video, write down the correct answer, and then as I discuss the answer, compare your answers to mine. If you have a different answer, please, or a different approach, please be welcome to also post that down in the comments. All right, let's get right into the questions. The diagram below represents part of a molecule. Study the diagram and answer the questions that follow. So they say that the diagram represents a part of the molecule, of a molecule. They did not specify which molecule that is. So it is your job to be able to identify the molecule by studying the diagram. Right, so we have a molecule there. Now let's check if we can identify what molecule this is, okay? This molecule has two strands. The first strand is there and the second strand is right over there. Now the molecule that we know contains two strands is DNA. Okay, so we are assuming that this is DNA, and the reason why we say it's DNA again is because we see that the two strands are joined together by weak hydrogen bonds, as you can check. So between the nitrogenous bases, we have weak hydrogen bonds that are joining the two strands. So one, I submit that is weak hydrogen bonds because it is joining the two strands of DNA at the point where the two nitrogenous bases of opposite strands join together. All right, so number one, I submit as weak hydrogen bonds. And then let's go to number three. As you can see, number three over there is attaching to the nitrogenous base, okay? So, which component of a nucleotide did we say attaches to a nitrogenous base? We said that it is the sugar portion. But because we said that this is a DNA molecule, we have identified this as a DNA molecule, we need to specify which type of sugar that is. So I submit that as deoxyribose sugar. And then the last part that is not attached to the nitrogenous base, that component of the nucleotide would be a phosphate group. We are also given a key there. So A there represents the nitrogenous base adenine, and G there represents the nitrogenous base guanine. Now that we have studied the diagram, let us look at the questions. Question number 1. Identify the molecule in the diagram for 1 mark. Like we said, because it has two strands and the weak hydrogen bonds are joining the two strands, this molecule has to be DNA. So you can write the word DNA, the abbreviation DNA, or in full, deoxyribonucleic acid. Question 1.2. Of course, you're going to find a question like this. Label the parts numbered 1, 4, and 5 respectively. Okay? So guys, an exam tip. Please do not just write the names of the parts. Please do not just randomly write the names of the parts. Like you'd write a part, comma, a part, comma, a part, comma. The question says respectively. So when you write the names, write 1 and say what 1 is. 4, say what 4 is. And 5, say what 5 is. Because at the end of the year, your papers are going to be marked at the marking center. Your teacher will not be marking your paper. And imagine, put yourself in the shoes of a marker. If a learner just wrote 3 random parts without mentioning which one is 1, which one is 4, which one is 5, it's going to be very frustrating for that particular marker, and they might mark you incorrectly. Okay, especially when the order of those parts are wrong. So please write 1 and tell us what 1 is. 4, tell us what 4 is. And 5, tell us what 5 is. So, when we were studying the diagram, we had already said it is weak hydrogen bonds. Let's see if that is correct. That is correct. So as you can see, 1 is weak hydrogen bonds. I have put the word weak in brackets because it is just an addition in terms of this answer. It is sufficient to just write hydrogen bonds, even if you did not include the word weak. But I advise you to write weak hydrogen bonds. I advise you to write it in full, okay? And then which one should we label as well? We should label part number 4. Okay, now part number 4, because it's on the inside of the strand, it is a nitrogenous base. But you don't just write nitrogenous base. Check the complementary base was specifically labeled as adenine. Okay, it was labeled as A. Therefore, you need to write the specific nitrogenous base that is complementary to A. Remember our analogy. The apple falls from the tree, and the car parks in the garage. Okay, therefore, we said that whenever you see an A, on the complementary strand, the nitrogenous base will be T. So for 4, is thymine the correct answer? Yes. So if you wrote T, that is correct. If you wrote it in full as thymine, that is also correct. And then 5. Let us check 5. So 5 is a nitrogenous base as well. But when you check on the complementary strand, when you check the complementary nitrogenous base, it is not labeled as anything. So where is your clue? Your clue is in the shapes of the nitrogenous bases. Now let us find a shape of a nitrogenous base that is similar to that shape over there. So this shape and that shape is the same. So if on this nitrogenous base having this shape is G, it is guanine. Therefore there it will also be guanine. All right, so you can slot it in and say that part is guanine. And what is complementary to guanine? That is cytosine. It means part labeled 5. This one over there would be cytosine. Okay. Following question. What is the collective name for the parts numbered 2, 3, and 4? Let us check. Part 2, we said it's a phosphate group. Part 3, we said it's a deoxyribose sugar. And 4, even though it is not put there, 4, it's a nitrogenous base. So a phosphate group, a sugar portion, and nitrogenous base forms what? It collectively forms a nucleotide. So I've said DNA nucleotide. So even if you just wrote nucleotide, the word DNA is in brackets. It's not compulsory for it to be part of your answer. So if you wrote nucleotide, that is correct. Following question. Label the part name numbered 3. The part numbered 3, there it is. We said that is the deoxyribose sugar. It is not enough for you to just write sugar portion, guys, because we had identified this molecule as a DNA molecule. You must specify that the sugar that is contained in the sugar portion at 3 is deoxyribose. All right? Okay. The following set of questions are on the next slide. So this is the second activity on on the structure of the cell. Study the sequence of nitrogenous bases below and answer the questions that follow. Let's look at the sequence of nitrogenous bases. Remember, the word sequence of nitrogenous bases, that means the order in which they appear. Okay? What is the first, second, third, fourth, fifth of nitrogenous bases? So the word sequence, it just simply means the order in which the nitrogenous bases appear. Okay, now the nitrogenous bases are put in triplets. A triplet is 3, right? Triplet means 3. Triplet does not mean 3 of the same. Okay, triplet just means 3, number, even though the individual items are not the same. I know with triplets, we often associate the word triplet with triplet children. And remember, triplet children may be identical and may not be identical. All right? So triplet just means 3. So in this case, it is 3 what? 3 nitrogenous bases in a particular sequence. So the sequence in which they follow, AGG is our first triplet, ATC second triplet, CCC third triplet, and AGC is our fourth triplet. All right, let's look at the questions. How many of the following are represented? Sub-question A of question 2.1, triplets of nitrogenous bases. So how many 3s, how many multiples of 3 do we have? The first triplet, like we said, is AGG. The second triplet is ATC. The third triplet is CCC. And then the fourth triplet is AGC. So these are triplets of nitrogenous bases. One triplet has 3 nitrogenous bases. So how many of these triplets do we have? The correct answer is 4. Okay. Sub-question B, nitrogenous bases. Okay. So how many nitrogenous bases are represented there? So we have 4 triplets of nitrogenous bases. But how many nitrogenous bases do we have in total? So each triplet contains 3. So this will be 3, 6, 9, 12. Or you can count them individually. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. So the correct answer is 12 nitrogenous bases. C, how many nucleotides? Okay, so this one can become a bit confusing. The reason is that here we are just given nitrogenous bases. We are not given the complete nucleotides. But remember, one nucleotide has a phosphate group and it has a sugar portion, and then on the inside it would have a nitrogenous base. Okay, it would have a nitrogenous base. So each nucleotide has one phosphate group, one sugar portion, and one nitrogenous base. So the nucleotide for this nitrogenous base, for this one over here, would contain cytosine there. For that one, would contain guanine there. For that one, it would contain guanine there. For that one, it would contain adenine there. So what I'm essentially saying here is that even though you are just given the nitrogenous bases, we know that nitrogenous bases do not travel alone. Okay? They travel with a sugar portion and a phosphate group. So even though we are just given the nitrogenous base, each nitrogenous base, there is only one nitrogenous base in each what? In each nucleotide. So if we have one nitrogenous base, we have that represents one nucleotide. Therefore, here we have 12 nitrogenous bases and we are going to have 12 nucleotides as well. All right. Following question. Guanine nitrogenous bases of the second strand of DNA. Let's read the question carefully. How many guanine nitrogenous bases of the second strand of DNA? Now remember, we said that we have two strands of DNA. One strand and the complementary strand. Okay? So, these 12 nitrogenous bases are contained on one strand. Okay? So, on the other strand of DNA, we would also have 12 complementary nitrogenous bases. All right. Now, we are on one strand, we are given 12 nitrogenous bases. Okay? And then the question is asking, how many guanines would we have in the second strand of DNA? Remember our analogy. Adenine goes with thymine because the apple falls from the tree. And then the car parks in the garage. Therefore, C goes with G. Okay, so the question is asking how many guanines are we going to find? What is guanine complementary to? Guanine is complementary to cytosine. So let's go to that strand and let us code for the complementary strand. So cytosine, on the second strand, we would have guanine. All right? Then we have 3 cytosines here. So it would be G, G, and G on the opposite strand. We have another cytosine there. So on the opposite strand, it would be another G. We have another cytosine there. On the opposite strand, it would be a G. Okay? So we count how many cytosines we have. And then on the complementary strand, there would be that many guanines. So we have 1, 2, 3, 4, 5, 6 cytosines. Therefore, on the complementary strand, the second strand, we would have 1, 2, 3, 4, 5, 6 guanines. Let's check if that's the correct answer. That is precisely the correct answer. If the above sequence of nitrogenous bases represented one strand, okay, so this sequence of this sequence of nitrogenous bases, if this represents one strand of DNA, write down the complementary sequence of bases that would represent the second strand from left to right. Okay, so they're asking us to code for the complementary strand. Okay, to code for the complementary strand. Right. Now, let's just write it down on the side. If one strand of DNA, so this strand has C, G, G, A, T, C, 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 and A, G, C. Okay? The complementary strand would code for, remember our analogy, apple falls from the tree, car parks in the garage. Okay, so the complementary strand, car, garage, garage, car, garage, car. All right. And then apple, tree, apple, and then car, garage. Okay. Then on the following one, in the following triplet, we have car, car, car. So we are going to have garage, garage, and garage. And then here we have apple. So we are going to have tree, then garage, car, and then car, we are going to have garage. So the complementary strand of the second strand of DNA would contain this sequence of nitrogenous bases. Let's check. If that's the correct answer. So the first triplet reads CGG. So the complementary triplet would be GCC. The second one, ATC. The complementary strand of that, the complementary triplet would be TAG. And then the third one is CCC. Then it would be GGG. And AGC would give us TCG. Okay, so that is the end of this activity. The last activity, guys, it's a bit more complicated. It's a cognitive level 4 activity, which requires you to apply your knowledge and do calculations. All right, let's look at the question. It says, if a nucleoplasm of a cell contains a total of 5,500 nitrogenous bases, so the total of nitrogenous bases is 5,500, okay? Of which 30% of the 5,500 are guanine. 30% of 5,500 are guanine. The question, calculate the number of each DNA base. Show all workings. All right. So guys, I am not very good at mathematics. The last time I did mathematics was in high school, all the way in 2009. I am not particularly great with mathematics. So there would be an easier way for you to calculate this. I just did it in a way that makes sense to me. Okay. There are many different ways to do calculations. There are many options to do calculations. So what I have on here is the way that I articulated it in my brain. Okay, so this was my thought process. I am given a total number of 5,500 nitrogenous bases. So all in all, there are 5,500. And what I know is that 30% of this 5,500 is guanine. Okay, so the first thing that I must do is calculate 30% of 5,500 to get how many of the 5,500 would be guanine. All right, so 30% is guanine, so I want the number. The question requires me to calculate the number, not percentages, but the number. So I firstly need to convert 30% into an actual number. All right, so let's check. Number of guanines would equals to 5,500 divided by 100 times 30%. And that gives me 1,650. So I've already gotten the number of what? Of guanine. So 30% is 1,650. Okay. This is how the formula that I use to convert percentages to numbers. I know there's probably another way for you to do it. It's fine as long as the correct answer is there. So, what do we also know? We know that the number of guanine nitrogenous bases equals to the number of cytosine nitrogenous bases. So if guanine is 1,650, cytosine is also 1,650. So we've attained the number of the two. Okay, so the number of G equals to the number of C. Therefore, if G is equals to 1,650, then C is also equals to 1,650. So we are left with two nitrogenous bases, because remember from the question, we need to calculate how many of each nitrogenous bases. So we are left with what? We are left with adenine, it is unknown. We are also left with thymine, that is also unknown. All right. Now, what do we do next? We need to determine the number of adenines and the number of thymines. All right. What we also know is that the number of adenine equals to the number of thymine. All right. So we need to subtract 1,650 and another 1,650 from the total 5,500. So 5,500 minus the number of guanines 1,650 and minus the number of cytosines 1,650. That is where our first mark is, equals to 2,200. Okay, so the nitrogenous bases that are left equals to 2,200. Or some people may want to add 1,650 plus 1,650, which is 3,300. And you say that 5,500 minus 3,300 also gives you 2,200. Okay, so from there, we are left with 2,200 nitrogenous bases. Then those 2,200 nitrogenous bases are of adenine and thymine. And like we said, the number of adenine equals to the number of thymine. Therefore, all we need to do is divide this remaining 2,200 by 2, and that would be the number of adenines and the number of cytosines. So 2,200 divided by 2 would give us 1,100. All right? So each remaining nitrogenous base, adenine and thymine, will equals to 1,100. So the final answer is G equals 1,650 and C equals 1,650. A is 1,100 and T is 1,100. Okay, that's where our final mark is. This final 2 marks, this is our compulsory answer, because remember, we needed to calculate the number. We needed to calculate the number of each nitrogenous base. That's what the question requires us to do. Okay, so for 2 marks, that is the final answer. And then we have any 2 of these calculations as additional marks for the instruction that says show all workings. Okay, so 2 for the compulsory mark. There we go. This asterisk represents the compulsory mark, and any 2 for the workings. That's where we get our 4 marks. Please comment on what was it, the thought process or the methodology that you employed when you're answering this question. Leave that methodology for me in the comments. I would highly appreciate that, because mathematics is not my thing. Maybe there was an easier way. I'd like to know, please, so that I can employ that in my classroom when I'm teaching next time. Okay, guys, we have reached the end of lesson 2 on the structure of DNA. I hope that this was very productive for you as it was for me. If there's any questions that you have, leave them in the comments. Any contribution that you need to make, leave them in the comments. And please remember to like the video, hit the subscribe button so that my channel will never be lost somewhere in the YouTube algorithm. And please do me a favor and share the link to this video with your friends, with your teacher, with your classmates, so that they also would benefit from these videos. So, thank you so very much for watching. The next video will be lesson number 3, and that will be based on DNA replication. All right? Please don't miss that video, and I will see you next time. Goodbye.